So we've gone over genetic change and we've gone over microbial control and growth. Let's talk about how these microbes actually cause disease. How do they get into our cells and how do they cause disease? So this is microbial infection and pathogenesis. All right, first of all, it's got to get into the host. That's the thing. In order to cause you a disease, it has to be able to get in. All right. So there's a couple definitions. Infection is when this microorganism is actually established and growing in a host, whether or not you're harmed. Okay. The disease is when there's actual tissue damage that starts to impair how your cells function. Pathogens, by definition, are these microbes that can cause disease or tissue damage. And pathogenicity is the ability of that microbe to damage. So some organisms are more pathogenic than others. Okay, so that's these definitions. All right, so first of all, this adherence is the ability of these guys to attach to your tissues. It's necessary. You have to be able to adhere somehow. And we're going to talk about this, but they gain access by a number of ways. Usually on the surface, if you have a cut, or particularly your mucous membranes, because you're breathing this stuff in, or something got penetrated, or something like that. All right. So what happens is you get this exposure. And this bacteria, you can see, it adheres to the skin or the mucosa, and it has this ability to tunnel through that epithelial layer. And then once it tunnels in, it can reproduce and grow and produces these, what we call virulence factors and toxins. And that's the infection process. And then once you get the disease process, is a lot of times your body is able to fight it off, but sometimes it's not. So the toxins can be local or they can be systemic. They can be very invasive, and you get tissue or systemic damage, and that's part of that disease process. Okay. So these organisms have a lot of different receptors coating them that allow them to attach. The tissues also have receptors. And this is where the bacteria virus binds. These are just, proteins or glycoproteins sticking up on the outside of these organisms, or they may have pili. We'll talk about it, but they're things that somehow that bacteria or virus can attach. And viruses attach differently than bacteria, but there's, we talk about bacteria have these hairs and stuff like that. And the cells have, glycolipids and glycoproteins that are sticking up that allow these guys to stick. So these pathogens, a lot of these organisms have adhesins that are glycoproteins or lipoproteins on the surface that allow it to form a chemical bond between the glycoproteins and the lipids on the surface of your cells. Okay, you can make a chemical bond. So here's an example. You got Staph aureus, and it's got this adhesin, and it's able to stick to this fibrinogen in the cell and allow it to get in. Okay. Some of these adhesive structures, capsules, bacteria capsules, have that thick coating on the outside, and that capsule serves two functions. It's sticky. A lot of them have slime layers and that sort of thing. They're sticky. They contain these receptors that allow them to attach to the tissue. Don't forget, we've got millions of years of evolution here. The capsules such as those on, I said strep pneumo, that's an encapsulated strain, it protects the bacteria from being ingested, because when these white cells try to grab a hold, they can't grab a hold of them, because the white cell is going to try and just go around and surround it and engulf it, phagocytize it. And these capsules make it hard for the cells to phagocytize these certain encapsulated strains. Okay. So you can see this capsule around strep pneumo, and it makes it harder for these cells, these white cells, to get a hold of these guys. All right, other things that help the bacteria attach, I mentioned already is pili, the fimbriae, those little short hairs, the flagella. They're on the cell surface. They function in attachment. The pili, I said, are typically longer and fewer in number than the fimbriae, but some pili function, we talked about already, these conjugation pilis. Flagella help because they allow the organism to move. A lot of these organisms can tunnel into tissue because they're motile. And that's, when we talked about this motility test that we did, looking at, we'll look at motility actually this week in lab, and you'll see that some organisms are motile. And because of their flagella, whatever, they have an ability to move from one place to the next. So that allows them to tunnel under the tissue. All right. All right. Once they get in, they colonize. And that is the growth of these organisms after they've gained access. Okay. This process begins at birth. You don't think about it, but when the baby is born, the baby is exposed to mother's bacteria, normal flora in the vaginal area, if it's a vaginal delivery, or when they, if it's a C-section, they're still going to go into a nursery or something that's, it's not sterile. So that process of colonization starts at the time the baby is born. And that's how we gain our normal flora, and that's how we begin that process. So typically it starts, with the mucous membranes, very packed epithelial cells, your thick epithelial cell layer coated in mucus and that sort of thing. But that's how you gain it. All right. All right. So now talk about how it gets in. You get this exposure. Let's suppose you get some staph on your arm. There's the staph. You're exposed to it. All right. There's your mucosal layer. There's your epithelial cells. And because of the staph, they can adhere to this tissue, begin to reproduce, and it can lead to a biofilm. We've talked about biofilms, that ability of these guys to stick together and cover an area. And when you get a biofilm formed, that's when you're really going to start to get invasion. Okay. So an example of that biofilm, I said, is your teeth, your cavities. It's your dental caries. That's the medical term for your cavities, are an oral microbial disease. When you get a cavity, it's because you've got, you've got a little area in your teeth and it's worn down, and these guys attach and form a biofilm called plaque, and the plaque builds up and you eventually get a cavity. And again, this biofilm is that slimy layer that you feel over your teeth when you get up in the morning. And that's why you go to the dentist and get the plaque removed, because if you can get that plaque removed, then that, helps you knock down these little lovely little organisms that cause dental cavities. Okay. particularly Streptococcus mutans, durans, sprinus. All right, so then we have to look at this virulence. All right. Again, it's the ability of a pathogen to cause disease. Some organisms are very, very virulent. Think about what's going on right now. Ebola virus, okay, hantavirus. Those are virulent, okay? So they have the ability to cause disease, and they have a very high ability. So you can talk about they have high virulence or low virulence. You know, some organisms just really, we can live with them and not a problem, but others make us really, really sick and we can die. So these pathogens use these different strategies to establish themselves, and they have toxins or destructive substances that they produce that indirectly or directly enhances how they can invade and how quickly they can invade and how much damage they can cause. This is an organ they want to get in. I would tell you most organisms, want to get in and be able to reproduce. They don't want to kill, because if you kill the host, they die too. So most of these guys are going to make you sick, but it's something that hopefully it doesn't kill you. We can measure virulence using the LD50, and it is the amount of the agent that kills 50% of the animals in a test group. And highly virulent pathogens, can be very, very high in the numbers that they are required to kill compared to 50%. I mean, if you look at Ebola, that's highly virulent. So you can look at this virulence. This makes more sense. Here's strep pneumo, okay? And you can see the percentage of mice killed, 100%, okay? But you don't have to inject very many cells into that mouse. Poor little mouse, bad part. Salmonella, not as virulent. It's going to take a lot more to kill 100%. So that's what they look at. The amount of agent kills 50% of the animals in the test group. And that's what they look at. So you're looking at this number right in here. Okay. And so this is the number of cells that's required to kill these mice. The other thing that we look at is the attenuation. And this is the loss of virulence. When you keep these organisms in a laboratory culture isolated from animals, they lose their virulence. It may be completely lost because they can't attach, they can't do those kind of things. We use attenuated strains of these pathogens, often in vaccines. Okay, you'll have an attenuated vaccine. We'll have a whole lecture on vaccines, but that basically means that those viruses have lost a lot of their virulence. Their LD50 factor has gone down. Okay, so that is important to us. So attenuation is like, here we go. You isolate the yellow fever virus from somebody that is diseased with it. It's no longer done this way, but this is just an example of it. And multiple passages of the virus to the rhesus monkey. And basically, then we put it in mouse embryos, and then we can do chicken eggs, and then we can administer the vaccine to somebody, and they will elicit an immune response and not get the disease. They'll be able to fight it because they know how to fight it. We don't make these anymore, but like this, but it's just an example of how they used to make the yellow fever vaccine in the past. Now we have more, biotechnology methods of doing this, but initially this is what we did. Okay. All right. Virulence in salmonella, you have once something that's interesting is that you can have what's called pathogenesis islands and plasmids. Now, salmonella, gives you a really nice diarrhea and other diseases. We'll talk about that later. It encodes for a large number of virulence factors. It allows it to directly invade, and these genes are clustered together on the chromosome. So they're all together in a little area on that chromosome. So we call this a pathogenesis island. One of the pathogenesis islands that salmonella has are a gene area that promotes more systemic disease, which contains its resistance plasmid. So you've got, not only have you got the pathogenicity, but you got the resistance plasmids to make it resistant to certain antibiotics and things. So, I mean, don't go into depth. I mean, I'm just trying to explain to you some of the things that can happen. I'm not going to test you on this, okay? But just understand. So here we're looking at our lovely little salmonella, and you can see the little endotoxin that's in the LPS layer that causes fever. There's products here. There's an endotoxin that causes diarrhea. There's fimbriae that allow it to attach. There's a virulence plasmid in here. I mean, if you keep looking at this O antigen inhibits phagocyte killing. So when the white cell tries to engulf it, it kills the white cell. There's your pathogenicity islands in the chromosome. So you look at it, these are all the things. This is just salmonella. These are all the things that these organisms have that allow it to cause disease. All right. Okay. A lot of times, the most important thing is, do you have a compromised host? Most of the time, if you're healthy, we can fight this off. We can do okay. But if you're compromised, you can really run into problems. And we'll talk about this again when we start going through diseases. But the pathogen-host interaction is dependent upon the health of the host and the health of the pathogen. So if you go and get a medical procedure, or you have an underlying condition like an immune disease, or you have cancer or something like that, that's going to predispose you to develop diseases. You go in and have surgery. Now we don't get it too much, but it does predispose you to post-op infections. Okay? Nosocomial infections, those are the infections that we as healthcare professionals cause when you go into a hospital and go in there and you get a procedure done and you get an infection. It is called a nosocomial infection. People are sick. People are coming into a healthcare facility because they have infections. So those organisms are in the healthcare facilities. The healthcare facilities try as much as they can to keep those at a minimum, but about 2 million people a year will get a nosocomial infection that they pick up in the hospital. And the infections with these viruses and stuff, that we talk about, surgery, HIV, immune, they'll weaken your immune system, and therefore they're going to have a problem. Opportunistic infections are those that by definition are caused by organisms that do not cause disease in healthy hosts. So if you're healthy, if you don't have a weakened immune system, those guys are not going to cause a problem. But because your immune system is weakened, those organisms are going to take the opportunity to infect you. Okay? And that's why we call them opportunistic infections. There's several ways that these organisms invade, but it requires the pathogen to break down your tissues. And they often use enzymes to do it. One famous one is hyaluronidase. Okay, I'm sure you've heard in ads about hyaluronic acid. Hyaluronic acid is really the glue, think of it, that holds cells together. Okay, and it allows these cells, your cells in your epithelium, to stick together. If you have an enzyme that breaks down that bond, then what happens is, and I'll show you a picture, what happens is that it allows little spaces between those cells, and that allows those organisms to get in. Okay. We also have coagulase and streptokinase. What's interesting is coagulase, particularly coag positive staph, it can get in and then it coagulates. It clots around itself, and it makes it very difficult for your immune system to see it, because everything around that staph is your cells. And then what happens is, and we'll talk about staph later on, we'll have a whole lecture on it, but what happens is that it divides and divides and divides and inside that little clot, and then it breaks it open and the staph go out and you've got a staph infection. Streptokinase is exactly opposite. It has the ability to break down those tissues. It's an enzyme that allows it to break down clots and tunnel through. So very different mechanisms. So here's your hyaluronidase. Okay, so here's your cell. There's your hyaluronic acid is between these cells, allows these cells to stick together and be safe. When you put strep on here, it has a hyaluronidase that gets secreted, and it allows it to break this hyaluronic acid down, and then it can tunnel through and go into your tissue layers below. Okay, and it gets in there and gets deeper. That's strep pyogenes. If you remember your beta strep, this is your strep throat. We'll get into that. Staph aureus, this is the catalase positive and coagulase positive staph. All right, so staph will be deposited, and as I mentioned, it forms a clot. And the clot walls this staph off, and the immune cells can't see it because it's in a clot protected by your own cells. And then what will happen is eventually it will open up and boom. And again, streptococcus has streptokinase. It will dissolve a clot and it'll release the pathogen into the bloodstream. So many different things that they can do. All right. Then a lot of them produce exotoxins and endotoxins, but they have a lot of toxins that they produce. Enterotoxins. These guys are very efficient, okay? So toxicity is the ability of these organisms to cause disease by the means of a toxin, okay? And we have three basic categories of exotoxins: cytolytic toxins, AB toxins, superantigen toxins. And I've just put a chart. I have to memorize these. Okay, but here's just some of the organisms. I just wanted you to see an example of some of the organisms and their very classic exotoxins and cytotoxins and what happens. Okay, so tetanus, Clostridium tetani. It has an AB toxin. We'll talk about what an AB toxin is, and it causes paralysis. Okay, all right. And you can see the Clostridium is notorious for this. because that's gas gangrene, botulism, Botox. This is Botox, Clostridium botulinum, but they give you it in tiny, tiny, tiny amounts. Okay. But you can see all the things that happen. E. coli, we'll talk about, it causes a nasty gastroenteritis. It's called a sugar toxin. Like there's a sugar toxin, but there's a sugar-like toxin, and this is what causes the bloody diarrhea. So it's the toxins being secreted that cause the problems in your body. And I went on and gave you a few more. Again, you don't need to memorize these, but you need to understand what these toxins are. So let's look at this. The AB toxins, these are some of the most well-known. They are very potent. And they are the ones that diphtheria, tetanus, botulism, cholera. We'll go through some of these diseases. And, some of them will block protein synthesis or whatever. But the AB toxin is named AB toxin because it is made up of the active domain and the binding domain. Okay. So let me go back here. So what the active domain promotes this enzymatic activity, okay, that modifies the intercellular target, modifies the target. And so, and it modifies it, it kills the cell. Yeah. The binding domain is exactly what it says. It helps the organism bind. So you have a toxin that allows the organism to bind and at the same time kill these cells that it's attaching to. That's the bottom line. Okay? All right. So neurological toxins, we looked at some of these. Some of these exotoxins, again, can block protein synthesis. You can have neurological exotoxins like botulism and tetanus toxins. Clostridium tetani, which causes tetanus, Clostridium botulism. It produces very potent AB exotoxins that, again, it's a neurological toxin. So they will produce neurotoxins, and these can kill you easily. I mean, you get a good case of botulism, and you've got a real problem. Same thing with tetanus. This is why you get your tetanus shots. Okay, these are not good diseases to get. So we'll talk about these more as we get into actual diseases and talking about some of these organisms. But I brought the botulinum toxin just to see, again, this is in Botox. And what happens is because it's a neurotoxin, you normally have muscle contraction, and hopefully you've had A&P, and you know that you've got these acetylcholine receptors on the end of the skeletal muscle. And what will happen is these normally it's released and you will be able to contract that muscle. Botox acts on this end of this motor plate and prevents the release of this acetylcholine. So the muscle cannot contract. Okay, and it is irreversible. So this is what makes botulism so bad. And you can see, I don't want you to go crazy on this, but you can see that you get this muscle relaxing. And you can read through this, but it can actually, the neuro, it's just, it becomes very flaccid. And it can cause the spastic paralysis. So it can go the other way. So again, I'm not going to test you on this, but I want you to just have an idea of some of the things. Normally you would have a relaxed muscle, and this can cause it to make spasms. Okay, so you can either cause a problem where you can't contract the muscle, or you can have problems where you are causing spasms. So it's not a fun thing. Exotoxins, enterotoxins are exotoxins that affect the gut, entero, okay, enteric. Generally, what you have is massive loss of fluid. Vibrio cholerae, which causes cholera. I'll show you how it works. Has an AB type exotoxin. And I will tell you the problem with cholera and why people die with cholera is that you can, I don't have pictures of it anymore, but you can lose 20 liters of fluid a day. You just get this watery diarrhea that doesn't stop. And, sorry. And when they have cholera epidemics, they used to have what are called cholera cots, where they literally put the patient on a cot with a hole cut where their bottom is and put a bucket underneath there to catch all the fluid that is just running out. And these people get so dehydrated and electrolyte imbalance, they die. Okay. But this is what it does. I mean, I'm not going to go into it, but I want to just show you that this is what happens, that toxin is produced and it elevates and blocks the sodium ions moving to the intestinal lumen. And so you get these massive amounts of water that move out, and you get this really nasty diarrhea that, I mean, you're just all the time just leaking fluid. All right, then you can get these cytolytic exotoxins, and this is the cytolytic cell lysine. They work by degrading the membrane, and they cause cell lysis. Some of these are hemolysis. They will rupture red blood cells. Okay. Staph has an alpha toxin that lyses red blood cells. We can see this on a blood agar plate. Okay. And then, we'll talk about this. We have alpha streps, strep pyogenes. That's strep throat. And, and so we will classify this according to the type of hemolysis we see. And we'll talk about beta hemolysis and alpha hemolysis. But so and the alpha toxin will cause this beta hemolysis, this complete destroying of the red blood cell, the release of the hemoglobin, and that's why it's so bad. But we're going to go into staphs and streps and talk about hemolysis and what happens with these a little later on in the course. But just realize that these guys can actually hemolyze blood cells. Okay, so you can see that little alpha toxin, and it gets into that cell membrane, and it can just start lysing cells. All right, then we have what's called superantigens, and these can cause the immune system to be overstimulated, and this is really what causes problems. It will cause the patient to go into shock and die. Generally, it's due to a localized infection, but the systemic effects from these organisms. So your gram-positive bacteria, Staph aureus that we just talked about, or strep pyogenes, your beta strep that has the hemolysin, they are major producers of superantigens. And poisoning can be triggered by food poisoning, particularly Staph aureus, toxic shock syndrome, pyogenic fever, really high fevers, but these guys can kill you if you don't get care quickly. Okay. You have to, I mean, that's why, we get pretty aggressive in certain times, and that's how people can die from these infections. Okay. Endotoxins, as we mentioned, and you look at this, think about this. This is an exotoxin because staph is gram positive, strep is gram positive. They are not gram negative organisms. So they have that thick layer of peptidoglycan. So that exotoxin is being produced by them. The gram negatives produce endotoxins, and that's found in that LPS layer of the gram negatives. So they are a structural component of that outer membrane of the gram negatives. Okay. And they are bound and released in toxic amounts only when the cells lyse. So I have some examples for you of the difference between an exotoxin and an endotoxin. I'm not going to go crazy on the test with this, but you need to understand that exotoxins, you can see they're often highly toxic, sometimes fatal. They're very specific. You can destroy them. Sometimes you can heat treat, but it's hard to kill the toxin. Okay. It's encoded in the extrachromosal elements of these organisms in the lysogenic bacteriophages for the viruses. Endotoxins, gram-negative bacteria. The symptoms generally much milder. They're moderately toxic. They're very rarely fatal. Okay. So that's really the big difference is they often produce a fever. Usually these exotoxins don't produce a fever, but the exotoxins will sometimes kill you. So just understand that they can do that. All right, so that's it. Again, we're going to go into a lot more detail when we start talking into these diseases and getting into these diseases, but this is just an overview of what's going on. Again, I'll give you, for the test, I'll give you a test review, so what you need to know. I don't expect you to memorize all this, okay? All right.